Welcome again. So now today we will be discussing about this innate responses to the viruses, where we will talk about the role of the interferon and NK cell. NK cell are also the part of your innate immune system, and interferons are also the part of the innate immune system. So talking about the innate response to the viruses is unique in that it is geared towards eliminating this intracellular pathogen. The two major mechanisms for dealing with the viral infection are interferon alpha and beta and NK cell. So talking about the interferon, what are the interferons actually? Interferons are the family of eukaryotic cell protein classified according to the cell of the origin. They are interferon alpha and beta, are produced by the variety of virus infected cell. They do the followings: act on the target cell to inhibit the viral replication, not the virus, and are not virus specific. So there is the two important point of this interferon. These are the protein, these are the eukaryotic protein, and if you understand about this protein interferon, they act on the target cell to inhibit the viral replication. Means that they actually act on the target cell to inhibit the viral replication, but not the viruses. That is important. If you see over this picture, you can see there is a cell that is infected with the viruses. The viruses attack that cell that has been infected. Now it is this cell has infected with the virus. They also release this interferon. This interferon comes into their environment and then act on the surrounding cells. So what they do in the surrounding cell, they inhibit them in this cell. They produce this antiviral protein, halts the virus production, and since this antiviral protein is there, there will no viral replication in this cell. Important thing is, this interferon is not acting on the virus, they are acting on the surrounding cell. That is important actually. So they act on the target cell to inhibit the viral replication, not the virus. So this target cell, they are acting on the target cell. They are inhibiting, they are actually causing changes in the surrounding cell, the targeted cell, so that the virus will not again infect the surrounding cell or nearby cell. So that is their target. Suppose I am a one cell, I have got infected, then I am already dying, but I will release this interferon, and this interferon will be dispersed into the surrounding, and all the surrounding target cell will be able to inhibit the viral replication. But you have to remember, this interferon cannot act directly on the viruses. They are acting on our own cell, our own surrounding cell. That is important. So they acts on a target cell to inhibit the viral replication, not the viruses. So they will act on our own surrounding cell to prevent from the viral replication or viral infection. That is the role of the interferon, and they are not virus specific. So this is response to any viruses. So if it is virus, if it is RNA virus, DNA virus, double stranded, single stranded, doesn't make differences. Any response, any virus has infect our cell, they will release this interferon, and response will be same for each and every viruses. It can't be specific, like one type of response for DNA virus, like herpes virus, another type of response for the orthomyxovirus, or say paramyxovirus, or say influenza or corona. Not that, like that. They are not virus specific. They do the responses, they show the responses similar to each and every viruses. The response is same. If the cell is infected, then the viral infected cell will produce the interferon that will goes into the surrounding cells, and the surrounding cell will be inhibited. The surrounding cell, what will do? The surrounding cell will have this will induce, what will happen? Interferon inhibit the viral protein synthesis by activation of the RNA endonuclease, which digests the viral RNA. So this interferon will enter inside this cell, and what it will do? It will actually, in this cell, what it will do? They will produce the antiviral protein that halts the viral production. So one function is they activate the RNA endonucleases in this cell. So this RNA endonuclease will digest the viral RNA once enter inside, and the viral replication will not take place, and this cell will not be infected with the virus. The another way is phosphorylation of the protein kinase. So there is the phosphorylation of the protein kinase, which inactivates this elongation factor 2, and that inhibits the viral protein synthesis. So the main aim is to prevent the surrounding cell to get infected from the virus. Suppose I am the viral infected cell, I have already got infected, I am dying, but I will release this interferon, this will goes to the surrounding, and my all neighbors, my all neighboring cell will be prevented or be prepared in such a way that it will not get infected with the viruses. And what are the preparation? They will activate that RNA endonucleases which digest the RNA, viral RNA, and so the viral RNA, if entered, that will get digested and there will be no replication and the cell will be prevented. There will be another process, that's phosphorylation of the protein kinase, which inactivates this elongation factor 2 that inhibits the viral protein synthesis. So in this way, now your surrounding cell are get protected from the viral infection. So this is the role of the interferon, and that is the part of your innate immune system. But you have to remember, this does not target the virus. They are not virus specific. The response is same to all the viruses, and they are not targeting the virus. They are actually targeting that cells, that's this cell, our own cell, that inhibit the viral replication. Okay. So interferon, talking about the clinical correlation, since the first description of this interferon almost 50 years ago, so a multitude dramatic immunology roles has been discovered for this group of the protein. As a group, interferon induce increase in the expression of the class 1 and 2 MSC molecule and augment the NK cell activities. They increase the efficiency of the presentation of the antigen to the both cytotoxic and helper cell population. Cloning of this gene encodes the interferon alpha, beta and gamma, which has been made it possible to produce sufficient amount to make this clinical practical. So they had 3 types actually, interferon alpha, beta and gamma. So alpha has well-known activity. This has the well-known activity and has been used in the treatment of hepatitis B and C infection. So this has the well-known viral antiviral activity, and that can be used for the treatment of this hepatitis B and C. They also can, within the cancer therapy, this interferon gamma has the promises in the treatment of hairy B-cell leukemia, chronic myelogenous leukemia and Kaposi sarcoma. So this interferon gamma, which we are talking about, they actually have this antiviral activity against hepatitis B and C infection. So it is used in the treatment of hepatitis B and C infection. Also, they can use in the cancer therapy, where they have the promising effect in the hairy B-cell leukemia, chronic myelogenous leukemia and Kaposi sarcoma. So this is the clinical correlation and clinical utility of your interferon alpha. Okay, talking about the interferon beta was the first drug shown to have the positive effect on young adult and with multiple sclerosis. Patients treated with interferon beta have enjoyed longer periods of remission and reduced severity of relapse. So this disease called multiple sclerosis, in the multiple sclerosis, this interferon beta has an important role for longer period of remission and reduce the severity of the relapse. So this is useful in multiple sclerosis patient. And there is another like under interferon gamma, which is used in the treatment of this chronic granulomatous disease. We have talked about this chronic granulomatous disease previously, where there is a deficiency of NADPH oxidase, which transport the oxygen and form the nascent oxygen or superoxide, which is converted into hydrogen peroxide, and then in the presence of the myeloperoxidase and the chloride form the HOCl and kill the bacteria. So if in chronic granulomatous disease there will be no substrate of this hydrogen peroxide or superoxide oxygen, then in that patient we cannot kill the bacteria, mainly in the catalase positive organism. So in that patient this interferon gamma can be given, which what will do, this molecule is a potent inducer of macrophage. This is a potent inducer of the macrophage activation and a promoter of the inflammatory response. So its application appears to be significantly reverse the CGD patient inability to generate the toxic oxygen metabolites inside the phagocytic cell. So we know chronic granulomatous disease, there is inability to generate the toxic oxygen metabolite inside the phagocytic cell. But when we give this interferon gamma in the chronic granulomatous disease, this actually reverse that process. There will be the generation of oxygen derived this toxic metabolite inside the phagocytic cell, and you can kill the bacteria by giving interferon gamma to the chronic granulomatous disease patient. So this is a promoter of inflammatory response, a potent inducer of macrophage activation, and that helps in the reversal of this NADPH oxidase, that's oxygen derived toxic material inside the phagocytic cell. Okay, so we know there is interferon alpha, beta and gamma. The side effect of the interferon therapy are fortunately mild and can be managed with the normal acetaminophen or say paracetamol. They induce headache, fever, chill, fatigue, and they diminish with the continued treatment. So you can simply give the paracetamol and the side effect can be managed. With the subsequent treatment, the side effect will be less. Okay. So we have discussed about this interferon. Now we can also we'll discuss about this NK cell. This NK cell is one of the important cell that is actually part of your innate immune system. What they will do, they are the member of the innate immune system, and they can kill the viral infected cell and tumor cell. We'll discuss in detail in the next lecture about it. They are very interesting in killing. They actually part of the immune system. They kill the virus infected cell and tumor cells, and they have the receptors by which they recognize the cell. If they have found the cell is not fully functioning and fully good well, then they kill that cell. So they just, if they found this face is not familiar, then they can kill that cell. So this is the way which NK cell actually kill the tumor cell or say viral infected cell. We can discuss about this cytokines that has been present over here. The cytokines actually cytokines involved in the innate immune system are the like interleukin 1, 6 and TNF. They are pro-inflammatory cytokines secreted by macrophage and macrophage. They act on different cells like interleukin 1 released from the macrophage act in the hypothalamus, cause fever, an endothelial cell cause expression of the ICAM, and on the liver causes stimulation of the production of acute phase protein like CRP. In the interleukin 6 released from the macrophage, target cell is liver, synthesis of the acute phase protein. TNF alpha released from the macrophage, attack target in the hypothalamus, produce fever. An endothelial cell cause increased expression of this ICAM that helps in the this chemotaxis, or they say you can transmigration of your phagocytic cell towards the infected site. Then liver produces this acute phase protein, neutrophil activation, tumor cell apoptosis, fat and muscle that cachexia due to this TNF alpha. Talk about the interleukin 8, which is released from the macrophage. Their target set is leukocyte. They cause chemotaxis of your this leukocyte. They induce adherence to the endothelium. So there is a upgradation, or you can say configurating change, which helps in the release of this integrins in the phagocytic cell. They cause chemotaxis and extravasation. Interleukin 12, which is released from the macrophage and dendritic cell, they act on the NK cell and helps in the production of interferon gamma. Interleukin 15 that also released from the macrophage and acts on the NK cell and cause the proliferation of your NK cell. Talking about the interleukin 18 that also released from the macrophage that and cause the synthesis of the interferon gamma. Talking about the regulatory cells, there are some interleukins like interleukin 10 that is released by the macrophage and dendritic cell. This acts on the macrophage and dendritic cell and that cause inhibition of the interleukin 12 production, decrease expression of the co-stimulatory molecules, and decrease class 2 MSC class 2 expression. So this interleukin 10 is one of the regulatory interleukins. There is the interferon type 1 interferons like interferon alpha and beta. They are released from the dendritic cell and macrophage. They acts on the all cell and NK cell. In all cell, what they will do, they transit inhibition of the protein synthesis, increase class 1 MSC1 expression. So we know that's interferon that inhibit the protein synthesis of the cell, by which any bacteria, any viruses which enter inside does not replicate. If they cannot replicate, what will happen? There will be no new virus formation and the surrounding cells can be prevented. So they also act on the NK cell and cause activation. There is an interferon beta which is not released from the dendritic or macrophage. They are released from the fibroblasts. They also act on the all cell and they cause the transient inhibition of the protein synthesis and increase class 1 MSC class 1 expression. They also act on the NK cell or cause activation. Then there is a TGF beta. This is released by the macrophage and lymphocyte. They have anti-inflammatory role. So I have told you about this macrophages. When there is the acute inflammation, neutrophil come, fight and try to resolve the infection, try to finish the war. But if they are not able to do that, they will call the macrophages. Macrophages will come and analyze the situation. If the infection has been resolved, if the infection has been shut down, now they will release this TGF beta and shut down or resolve all the infection, all the inflammatory process. If the war is win. If their war is not win, if the infection is still persisting, the neutrophils has not able to clear the infection. If they have win the war, then what will do? Macrophage will release this anti-inflammatory product, that is TGF beta, and resolve the infection, resolve the inflammation. But if their infection is persistent, then this will go and fight the war. Then they will also process that antigen and go to the adaptive immunity, that is T cell, and take the help, and then again activate the formation of antibody, and then the chronic inflammation persists. So in this way, this cytokines of the immune system works. Let's end over here, and then we'll discuss about next chapter of NK cell subsequently. Thank you.